Welcome to our deep dive into data integration techniques in systems biology. In today's world, biological research generates vast amounts of data from various sources: genomics, proteomics, metabolomics, and more. Integrating these diverse datasets is crucial for gaining a holistic understanding of biological systems. But how do scientists bring together such different types of information? In this video, we'll explore the main techniques used for data integration in systems biology, why they matter, and how they help researchers uncover new insights. Let's begin by understanding what data integration means in this context. Data integration in systems biology refers to the process of combining data from different experimental sources to create a unified view. This is essential because biological systems are complex and interconnected. By integrating data, researchers can identify patterns and relationships that would be impossible to see in isolated datasets. For example, linking gene expression data with protein interaction networks can reveal how genes and proteins work together in health and disease. This unified approach is the foundation for systems-level understanding. Why is data integration so important in systems biology? The answer lies in the complexity of living organisms. Each type of biological data, whether it's DNA sequences, protein levels, or metabolic profiles, offers a unique perspective. However, no single dataset can capture the full picture. By integrating multiple data types, scientists can build comprehensive models that reflect the true nature of biological systems. This leads to more accurate predictions, better understanding of diseases, and more effective therapies. Let's take a closer look at the types of data commonly integrated in systems biology. These include genomics data, which provides information about DNA sequences; transcriptomics, which measures gene expression; proteomics, which looks at protein abundance and interactions; and metabolomics, which analyzes small molecules involved in metabolism. Each of these data types is generated using different technologies and has its own format and scale, making integration a challenging but rewarding task. Integrating biological data isn't straightforward. One major challenge is the heterogeneity of data sources. Different experiments produce data in various formats, scales, and qualities. For example, gene expression data might be continuous, while protein interaction data could be binary. Additionally, missing values and noise can complicate the process. Overcoming these challenges requires careful preprocessing, normalization, and the use of robust integration techniques. Before integration can happen, data must be preprocessed and normalized. Preprocessing involves cleaning the data, handling missing values, and correcting errors. Normalization ensures that data from different sources are comparable, often by scaling values to a common range or distribution. These steps are critical for minimizing biases and ensuring that integrated analyses are meaningful and reliable. Now let's explore the main techniques used for data integration in systems biology. These methods can be broadly categorized into three groups: early integration, intermediate integration, and late integration. Each approach has its own advantages and is suited to different research questions. In the following scenes, we'll break down each technique and discuss how they work in practice. Early integration, also known as data-level integration, involves combining raw data from different sources before any analysis is performed. This approach requires that all datasets be compatible in terms of format and scale. Early integration is powerful because it allows for the discovery of patterns that span multiple data types. However, it can be challenging due to the need for extensive preprocessing and normalization. Intermediate integration, or feature-level integration, combines data after initial processing but before final analysis. In this method, features are extracted from each dataset, such as gene expression levels or protein abundances, and then merged into a single feature matrix. This approach balances the need for compatibility with the flexibility to handle different data types. It's widely used in machine learning applications within systems biology. Late integration, or decision-level integration, involves analyzing each dataset separately and then combining the results. For example, separate models might be built for genomics and proteomics data, and their predictions are integrated at the end. This approach is useful when datasets are very different or when it's difficult to merge them directly. Late integration can help preserve the unique characteristics of each data type while still providing a unified outcome. Selecting the appropriate integration technique depends on the research question, the nature of the datasets, and the desired outcome. Early integration is best when datasets are similar and well-aligned. Intermediate integration offers flexibility for diverse data types, while late integration is ideal when datasets are highly heterogeneous. Researchers often experiment with multiple approaches to find the best fit for their specific needs. Statistical techniques play a key role in data integration. Methods such as principal component analysis (PCA), canonical correlation analysis (CCA), and clustering are commonly used to reduce dimensionality and identify relationships across datasets. These tools help researchers uncover hidden patterns and make sense of complex, high-dimensional data. Machine learning has revolutionized data integration in systems biology. Algorithms such as random forests, support vector machines, and neural networks can handle large, complex datasets and learn from multiple data types simultaneously. These methods are particularly useful for predicting disease outcomes, identifying biomarkers, and modeling biological networks. Network-based approaches are widely used in systems biology for data integration. In these methods, biological entities such as genes, proteins, and metabolites are represented as nodes, while their interactions form the edges of a network. By integrating different types of data into a single network, researchers can study the relationships and dynamics of entire biological systems. Ontologies provide a structured vocabulary for describing biological concepts and relationships. Ontology-based integration uses these frameworks to align data from different sources, ensuring consistency and interoperability. For example, the gene ontology helps standardize gene function annotations, making it easier to integrate and compare datasets from different studies. Databases play a crucial role in data integration by providing centralized repositories for biological data. Integrating data from multiple databases requires mapping identifiers, resolving inconsistencies, and merging records. Tools like Biomart and UniProt facilitate this process, enabling researchers to access and analyze integrated datasets efficiently. Semantic integration focuses on the meaning of data, using metadata and ontologies to link related concepts across datasets. This approach enhances data interoperability and enables more sophisticated queries. For example, semantic web technologies allow researchers to connect gene, protein, and disease information in meaningful ways. Ultiomics integration combines data from genomics, transcriptomics, proteomics, and metabolomics to provide a comprehensive view of biological systems. This approach enables researchers to study how changes at the DNA level affect gene expression, protein function, and metabolic pathways, leading to deeper insights into health and disease. In cancer research, data integration has been instrumental in identifying new biomarkers and therapeutic targets. By combining genomic, transcriptomic, and proteomic data, scientists can uncover the molecular mechanisms driving tumor growth and resistance to treatment. Integrated analyses have led to more personalized and effective cancer therapies. Data integration is also vital in studying infectious diseases. By merging data from pathogen genomics, host immune responses, and clinical outcomes, researchers can track disease outbreaks, understand transmission dynamics, and develop targeted interventions. This integrated approach has been especially important during global health crises. Visualization tools are essential for making sense of integrated data. Software like Cytoscape and R's ggplot2 allow researchers to create interactive network diagrams, heat maps, and other visualizations that reveal patterns and relationships in complex datasets. Effective visualization helps communicate findings and guide further analysis. Several software platforms support data integration in systems biology. Tools like Galaxy, KNIME, and Taverna provide user-friendly interfaces for combining, analyzing, and visualizing data from multiple sources. These platforms streamline workflows and make advanced integration techniques accessible to a broader range of researchers. Cloud computing has transformed data integration by providing scalable storage and computational resources. Researchers can now integrate and analyze massive datasets without the need for expensive local infrastructure. Cloud platforms also facilitate collaboration and data sharing across institutions and countries. Standardized data formats and protocols are essential for successful integration. Initiatives like the Minimum Information About a Microarray Experiment (MIAME) and the Systems Biology Markup Language (SBML) ensure that data can be easily shared and combined. Adhering to standards reduces errors and improves reproducibility. Quality control is a critical step in data integration. Researchers must assess the reliability and consistency of each dataset before combining them. Techniques such as outlier detection, cross-validation, and benchmarking help ensure that integrated analyses are robust and trustworthy. Ethical issues are increasingly important in data integration, especially when dealing with human data. Privacy, consent, and data security must be carefully managed. Researchers follow strict guidelines to protect sensitive information and ensure that integrated datasets are used responsibly. Personalized medicine relies heavily on data integration. By combining genetic, clinical, and lifestyle data, doctors can tailor treatments to individual patients. This approach leads to more effective therapies and better health outcomes, demonstrating the real-world impact of integration techniques. Drug discovery is another area where data integration shines. By merging chemical, biological, and clinical data, researchers can identify promising drug candidates and predict their effects. Integrated analyses accelerate the drug development process and reduce the risk of failure in clinical trials. In agriculture, integrating genomic, environmental, and phenotypic data helps improve crop yields and resistance to disease. Systems biology approaches enable breeders to select the best traits and develop more resilient plants, contributing to food security and sustainability. Ecologists use data integration to study complex ecosystems. By combining data on species, environmental factors, and interactions, they can model ecosystem dynamics and predict responses to changes such as climate shifts or habitat loss. Integrated approaches are vital for conservation efforts. Microbiome research benefits greatly from data integration. By merging genomic, metabolic, and environmental data, scientists can understand how microbial communities interact with their hosts and environments. Integrated analyses reveal the roles of microbes in health, disease, and ecosystem functioning. Neuroscience is another field where data integration is making a difference. By combining brain imaging, genetic, and behavioral data, researchers can unravel the complexities of brain function and disease. Integrated approaches are leading to breakthroughs in understanding neurological disorders. Evolutionary biologists integrate genomic, fossil, and ecological data to study the origins and diversification of life. By combining evidence from multiple sources, they can reconstruct evolutionary histories and understand the processes driving adaptation and speciation. Synthetic biology relies on integrating data from genetics, engineering, and computational modeling. By merging these diverse datasets, scientists can design and build new biological systems with desired properties, paving the way for innovations in medicine, energy, and materials. Environmental scientists integrate data from sensors, satellite imagery, and field studies to monitor and model environmental changes. This integrated approach supports decision-making in areas such as pollution control, resource management, and climate change mitigation. Public health researchers use data integration to track disease outbreaks, evaluate interventions, and inform policy decisions. By combining clinical, demographic, and environmental data, they can identify risk factors and design more effective public health strategies. Pharmacogenomics studies how genetic variation affects drug response. Integrating genomic, clinical, and pharmacological data helps identify which patients will benefit from specific treatments, reducing adverse effects and improving outcomes. Proteogenomics combines proteomic and genomic data to improve gene annotation and discover novel proteins. This integrated approach enhances our understanding of gene function and regulation, leading to new insights in biology and medicine. Metabolomics integration involves combining metabolic data with genomic and proteomic information. This approach helps researchers understand how genetic and protein changes affect metabolism, providing a more complete picture of cellular processes. Plant biologists integrate genomic, transcriptomic, and phenotypic data to study growth, development, and stress responses. Integrated analyses help identify genes and pathways that control important traits, supporting crop improvement and sustainable agriculture. Animal studies benefit from integrating data on genetics, physiology, and behavior. This comprehensive approach helps researchers understand animal health, development, and adaptation, with applications in veterinary medicine and conservation. Studying human disease requires integrating data from genetics, clinical records, and environmental exposures. This holistic approach uncovers the complex interactions that contribute to disease risk and progression, guiding prevention and treatment strategies. Future of data integration in systems biology is bright. Advances in artificial intelligence, big data analytics, and high-throughput technologies will enable even more sophisticated integration methods. As datasets grow larger and more complex, new tools and standards will be needed to keep pace. Sum up, data integration is a cornerstone of systems biology, enabling researchers to combine diverse datasets for a more complete understanding of life's complexity. From early to late integration and from statistical to machine learning methods, each technique offers unique advantages. Integrated analyses are driving discoveries across biology, medicine, and beyond. Thank you for joining us on this journey through data integration techniques in systems biology. As technology advances, the ability to integrate and analyze complex biological data will only become more important. By mastering these techniques, researchers can unlock new insights and drive innovation in science and medicine. Stay curious and keep exploring the world of systems biology.